So good morning to everybody. Today I am going to discuss about cell cycle. Cell cycle is a series of events that takes place in a cell as it grows and divides into two daughter cells. Purpose of the cell cycle in higher organisms. It maintains normal metabolic functions. It helps in reproduction and gametes formation. It helps in repairing and regeneration of damaged cells. Cell cycle consists of two main stages. One, interphase. Two, mitotic phase. Interphase has three stages. One, G1 stages, S phase and G2 stages. M phase having only one stage, that is called mitotic division. Interphase has three stages: G1 phase or gap 1 phase, S phase or synthesis phase, and thirdly G2 phase. Interphase is the longest phase in the cell cycle. In interphase, cell grows, replicates its chromosomes and prepare for cell division. First, G1 phase. During this phase, cell is metabolically active and continues to grow without replicating its DNA. It takes place in the cytoplasm and nucleus. In this phase, cells mature and increases in size. The duration of this phase is 8 to 11 hours. Cellular contents are duplicated except the chromosomes. Proteins are produced in G1 phase. DNA polymerase are synthesized for DNA duplication. S phase or synthesis phase. In this phase, the cell synthesizes and double its DNA. During doubling of DNA, the number of chromosomes remains constant. Histone proteins are wrapped around the DNA copies to make it stable. More phospholipids are produced in S phase. The phospholipids which make up the cell membrane and the cell membranes of the cell organelles. It takes place 6 to 8 hours. G2 phase or gap 2 phase. During this phase, the cell continues to grow and prepare for cell division. Mitochondria will divide and continue to grow until the start of metaphase. Checking of errors in DNA takes place in G2 phase. The time duration of G2 phase is 4 hours. Next, M phase or mitosis. It is divided into 4 stages. Number 1, prophase. Number 2, metaphase. Number 3, anaphase. Number 4, telophase. This M phase takes place nearly 1 hour. First phase is called prophase. In prophase, chromosomes are condensed and became visible. Each chromosome consisting of 2 chromatids, nucleolus and nuclear membrane disappears. So here that nucleolus and then nuclear membrane, it's not continuous, it is going for disappearance. Second phase is called metaphase. In metaphase, synthesis of spindle fibers takes place. Spindle fibers are attached to centromere of each chromosomes. The chromosomes are thick, coiled, or lined up in the center of the cell. So here that chromosomes are lined up in the center of the cell on the metaphase plate. The spindle fibers are originate in the opposite poles, so they are get attached on the each chromosomes of the centromere. So this will be the centromere. Here that spindle fibers get attached to the spindle fibers. Anaphase. In anaphase, centromere split and chromatids separate. Chromatids move to opposite poles due to shortening of spindle fibers. So here that the centromeres of each chromosomes get split and move to the opposite directions. Finally, telophase. In telophase, chromosomes moves to opposite poles. Nuclear membrane forms around the chromosomes. Nucleolus, Golgi complex and endoplasmic reticulums are reformed. Here the cytoplasm may be dividing. So if you see that the cell that there will be a reappearance of nucleolus and then nuclear membrane also takes place. Thank you.